Gregor Mendel presented his work outlining his discrete particulate inheritance model at two scientific meetings in 1865 in Brünn. While those presentations were positively reviewed in local newspapers, the scientific community all but ignored his findings, either because they did not understand the findings or just completely discounted them outright. He published his findings in a relatively obscure scientific journal in 1866, but over the next 35 years, Mendel's paper was only cited 3 times. The strong acceptance of blending inheritance, in which phenotypes of offspring are an intermediate of their parents, was overwhelming, causing Mendel's discoveries to lament in the scientific abyss. It was not until the turn of the 20th century that Mendel's discoveries were truly appreciated. In the 1890s, Dutch botanist Hugo de Vries began hybridization experiments of different plant varieties, including 15 different species. Unbeknownst to him, he was replicating Mendel's pea experiments. By the turn of the century, the results of his experiments led him to discover what are now known as the principles of Mendelian genetics. During his experimentation, de Vries was unaware of Mendel's work, but that was about to change. During the same decade, German botanist Karl Correns was also experimenting with plant hybridization. In his experiments with hawkweed, he, like de Vries, discovered the underlying principles of Mendel's experiments without previous knowledge of Mendel's work. However, as Correns was preparing his manuscript toward the end of the century, his literature search led him to Mendel's paper. Realizing that he had rediscovered the same principles he had developed in his own experiment, Correns reconfigured his manuscript as a rediscovery paper used to support and reintroduce Mendel's study published 35 years earlier. Shortly before Correns published, de Vries beat him to the punch. In his paper on the law of hybrid disjunction, de Vries provided experimental evidence allowing him to arrive at many of the same conclusions Mendel had come to. He had rediscovered the presence of dominant and recessive traits, as well as the 3-to-1 phenotypic ratio found in the F2 generation, and independent assortment. However, de Vries did not mention nor cite Gregor Mendel's work. The day after he had read de Vries' paper, Correns mailed his manuscript to be published so he could claim dual priority of the rediscovery of Mendelian genetics. The most significant difference between these two papers was that Correns had read Mendel's work and had couched his findings within Mendel's theoretical framework. Correns was furious. He realized that de Vries was attempting to lay the foundation for what is now known as genetics, without giving proper credit to the obscure German monk that originally discovered these principles. De Vries claimed his innocence by simply stating that he knew nothing of Mendel's work. Fair enough, but was it true? Based on an analysis of de Vries' writing, it appears that he was truly ignorant of Mendel's work prior to his first 1900 publication. Before 1900, de Vries did not use the terms dominant or recessive. Rather, he referred to those traits as active or latent. But in his 1900 publication, he referred to them with Mendelian terminology. Coincidence? Maybe. Secondly, de Vries claimed to discover the 3-to-1 phenotypic ratio in the F2 generation, leading to the principle of segregation. However, his actual results were grossly inconsistent with this. Some examples were closer to a 2-to-1 ratio, such as 99 hairy to 54 non-hairy variants, and others like flower color were closer to 4-to-1. Having ratios range from 2-to-1 to 4-to-1 would not have, arguably, led de Vries to independently recognize the principle of segregation. This suggests that de Vries was most likely just recently familiar with Mendel's work at the time of writing his initial 1900 paper, but sought to keep Mendel's obscure results suppressed, seeking to reap fame as the discoverer of these genetic principles. Correns not only properly reintroduced the world to Gregor Mendel, he added additional theory. While Mendel was the first to describe hybrids with 2 letters, such as big A, little a, he notated that true breeding pure lines from self-pollination with a single letter, big A for the dominant trait and little a for the recessive. Correns reinterpreted his and Mendel's results as the presence of a pair of heredity units, which we now give the genotypes, big A, small a, and little a, little a. Correns was the first to make the now famous 9-to-3 to 3-to-1 ratio predicted in the F2 generation, merging the principles of segregation and independent assortment into a single experiment. Corinz was also the first to link the concept of segregation with meiosis, a concept not understood in Mendel's time. Neither de Vries nor Mendel postulated that sex cells, such as pollen and eggs, housed one unit of heredity while vegetative cells contained two. Many other researchers were working in the field of hybridization at the same time as de Vries and Corinz. After reading the 1900 papers reintroducing the world to Mendel, British zoologist William Bateson became one of the leading defenders of the emerging field of genetics, a term which he coined, from the Greek word for descent. In his book, Mendelian Principles of Inheritance, published in 1904, Bateson translated Mendel's text for an English audience and argued that Mendel's principles would be generalizable to all plants and animals as well. In his writings, Bateson also coined some of the now ubiquitous terms of Mendelian genetics: allelomorph, subsequently shortened to allele, zygote, homozygote, and heterozygote. Following the rediscovery of Mendelian genetics, inheritance research exploded. The next major discovery has become known as the chromosomal theory of inheritance. American biologist Walter Sutton began studying inheritance of grasshoppers at the turn of the century. In 1902, he published a paper arguing that chromosomes occur in matched pairs, with one chromosome from each pair originating from that organism's parents. In 1903, Sutton expanded his theory to conclude that each pair of chromosomes segregates during the production of sex cells, such as sperm or egg cells, via meiosis. In other words, each sex cell, known as gametes, contain a full set of unpaired chromosomes. Sutton posited that during meiosis, the paired chromosomes aligned randomly and separated. This means that each gamete would contain a random assortment of maternal and paternal chromosomes, explaining Mendel's principle of independent assortment. During fertilization, the randomized assortment of unpaired maternal and paternal chromosomes of one gamete, such as sperm, fuse with the random assortment of another individual's chromosomes, creating a genetically unique offspring. It is important to note that the chromosomal theory of inheritance is co-credited with the German biologist Theodor Boveri, who determined that it's necessary for all chromosomes to be present for successful embryonic development of his model organism, the sea urchin. Furthermore, he discovered that sperm and egg contribute the same number of chromosomes during fertilization. These realizations occurred before Sutton's 1902 and 1903 publications. However, he did not link those findings to Mendelian principles until a 1904 publication. Interestingly, Boveri was the first to predict that cancerous cells were caused by abnormal chromosomes, a highly provocative hypothesis during his time. During Mendel's time, the mechanism of meiosis was not known, but understanding the specifics of meiosis proved instrumental in explaining his results. Mendel's principles of segregation asserts that an organism has two alleles, Mendel called them particles, for each genotype. A genotype determines the physical characteristic, phenotype, such as flower color. Homozygous dominant and heterozygous genotypes express a dominant phenotype, whereas homozygous recessive genotypes express the recessive phenotype. Additionally, Mendel suggested that the offspring of a mating will only receive one particle, or allele, from each parent, and that the probability of getting a specific allele is 50%. In other words, during meiosis, each gamete has an equal probability of acquiring an allele of a specific gene. He concluded that the consistency of the phenotypic ratio in the F2 generation, 3 dominant to 1 recessive, is only possible if each gamete has an equal probability of acquiring one allele over the second. But how? A diploid organism, notated as 2N, like pea plants and humans, have 2 copies of each chromosome, known as homologous chromosomes. In humans, one homologous chromosome comes from the mother and the other comes from the father. Each chromosome consists of several alleles. Homologous chromosomes have the same sequence of alleles. However, the alleles can differ between the homologous chromosomes. Therefore, on one homologous chromosome, you can have a dominant allele, while on the other, you can have a recessive allele in the case of a heterozygote. Or both chromosomes could have the same allele, as is the case for a homozygote. In interphase prior to meiosis, all chromosomes are replicated. In meiosis 1, homologous chromosomes pair up and then split or segregate, and eventually migrate to 2 different cells. Each cell at the end of meiosis 1 has either one replicated homologous chromosome or the other. In meiosis 2, the replicated homologous chromosomes split again, generating the gametes, which have one homologous chromosome for each chromosome pair. Gametes have half the number of chromosomes as normal body or somatic cells. The splitting of the homologous chromosomes explains the 3-to-1 ratio of the F1 generation of Mendel's monohybrid cross. In other words, sex cells have a 50% chance of receiving one chromosome or the other for each pair of homologous chromosomes, hence the chromosomal theory of inheritance. Mendel and de Vries both discovered phenotypes they were studying sorted independently. For example, purple-flowered pea plants can either have green seeds or yellow seeds, while white-flowered plants can also have green or yellow seeds. In other words, flower color was not dependent on seed color. With his 9:3 to 3:1 phenotypic ratio in the F2 generation, Corin's was the first to propose specific predictions for independent assortment, but he was not aware of the mechanism. The developing understanding of meiosis helped us determine how physical traits were independently assorted. With the emerging understanding of chromosomes, Walter Sutton realized that alleles coding for the different physical traits or phenotypes Mendel and Corin's were observing were located on different chromosomes. During meiosis, a pair of similar chromosomes, the homologous chromosomes, split during anaphase 1 and end up in one of two daughter cells at the end of meiosis 1. Therefore, each daughter cell at the end of meiosis 1 contains one of each of the original homologous chromosomes. However, the daughter cells, which eventually become the gametes, don't necessarily have the complete set of the original paternal chromosomes, nor the complete set of maternal chromosomes. Gametes end up with a random set of paternal and maternal chromosomes. For example, typical human body cells are diploid, 2N, composed of 46 chromosomes, notated as 2N equals 46. A set of 23 chromosomes originated from the father and 23 from the mother. During fertilization, these sets fuse, forming a zygote, the first human body cell. As your body develops, each body cell is composed of these sets of paired homologous chromosomes. When humans reach reproductive maturity, they undergo meiosis to produce gametes, either sperm or egg. During this process, a diploid cell, 2N equals 46, divides the number of chromosomes in half, which then migrate to opposite cells, becoming haploid, containing 23 chromosomes, N equals 23. Each gamete contains one of the original homologous chromosomes for each pair of chromosomes. Independent assortment occurs because the probability of any given gamete receiving either the original maternal or paternal chromosome is 50%. This means that one human can produce 2 to the 23rd or 8,388,608 different chromosome combinations. This explains why Corin's detected independent assortment with his consistent 9:3 to 3:1 phenotypic ratio in the dihybrid cross of the F2 generation. It just so happened that the phenotypes he was investigating were on different chromosomes. He got lucky. Sutton interpreted this result to indicate that the genes coding for Mendel's and Corin's different phenotypes were on different chromosomes, and he inferred that the chromosomes were the units assorting independently, not the individual alleles predicted by Mendel. Chromosomes are the unit of inheritance. While Bovary and Sutton hypothesized that Mendel's principles were related to meiosis, they didn't have experimental evidence to support their claim. That evidence came from the lowleaf fruit fly, Drosophila melanogaster. Beginning in 1908, American biologist Thomas Hunt Morgan began studying the fruit fly with the hopes of understanding inheritance in animals. At the time, Morgan dismissed both Darwin's theory of natural selection and Lamarck's inheritance of acquired characteristics. Rather, he sought to develop experimental evidence to prove de Vries' mutation theory as the basis of evolution. In this rival theory, de Vries speculated that new species were created nearly instantaneously solely by the mutations of the chromosomes. In contrast, Darwin and Lamarck argued that evolution was a gradual process involving selection pressures on a continuum of traits. While they differed in their mechanism, both used the concept of survival of the fittest to explain the specifics of their respective theories. In both of these theories, change happens gradually over generations. In contrast, mutation theory predicted that new species arise from mutations instantaneously and at random. Unlike plants, animals cannot be self-fertilized. He chose the fruit fly for three main reasons. First, they're easy to keep alive. You just give them fruit. Second, a fruit fly has an extremely short generation time, about 10 days. So data from many generations can be accrued in a relatively short amount of time. Third, fruit flies have a tremendous amount of phenotypic variation. Along with Fernandez Payne, Morgan began creating mutants of the fruit fly through chemical or radiation exposure. By 1909, heritable mutants began emerging. In 1910, Morgan discovered a white-eyed male mutant, where all the other fruit flies had red eyes. Flies with the original phenotype are called the wild type, red eyes in this case. He inferred that the white eye color was a mutation in the chromosomes, and then designed a set of crosses in order to determine how eye color was inherited. Morgan was shocked to find that the results of the F1 generation did not match Mendel's predictions. When he mated a white-eyed female with a red-eyed male, he found that all the females of the F1 generation had red eyes, while all the males of the F1 generation had white eyes. But how? Mendelian genetics would have predicted a 3-to-1 ratio of dominant to recessive phenotypes, independent of sex. During Morgan's time, the understanding of chromosomes was still emerging. As microscopy improved, enhanced visualizations of chromosomes led scientists to discover the sex chromosomes. In 1905, Americans Nettie Stevens and Edmund Wilson independently discovered chromosomal XY sex determinism. They discovered that males in animals have 2 homologous chromosomes that determine the animal's sex, X and Y. The X chromosome looks like an X when it condenses during meiosis, while the Y appears to look more like a V or a Y. A male has one of each of these sex chromosomes, XY. Females lack a Y chromosome, having 2 X chromosomes, XX. The discovery of the X and Y chromosomes led Morgan to speculate that the gene controlling for eye color of his fruit flies was on the sex chromosome, specifically the X chromosome. When a gene is located on a sex chromosome, it is said to be sex-linked. Hunt suggested that males expressing the white-eyed phenotype, a recessive trait, only need to have a single allele in order to be expressed, if it is on the X chromosome. The Y chromosome does not have a gene that codes for the proteins responsible for eye color. Since X and Y are homologous, a wide-eyed male would have the genotype XeY. Since the wide-eyed phenotype is a recessive characteristic in flies, he suggested that female fruit flies must have a recessive allele for eye color on both X chromosomes, XeXe. If this were true, he should be able to predict the phenotypic ratio of controlled crosses according to Mendelian principles. For example, if a heterozygous red-eyed female, XeXe, is crossed with a red-eyed male, XeY, the expected genotypic and phenotypic ratio can be determined using a Punnett square. The expected genotypic ratio would be 1XeXe to 1XeXe to 1XeY to 1XeY, which gives the phenotypic ratio of 2 red-eyed females to 1 red-eyed male to 1 white-eyed male. Let's look at another example. Construct a cross between a white-eyed female and a red-eyed male. Notice, you weren't given the genotypes, but with this information, we know what they are. Since white eyes is a recessive phenotype, we know that the female genotype is XeXe. Furthermore, since males can only have one X chromosome, whatever allele is on that chromosome is expressed. Therefore, his genotype must be XeY. If you construct a Punnett square for this cross, Xe times XeY, the genotypic ratio is 1XeXe to 1XeY, represented by 1 red-eyed female to 1 white-eyed male. With his investigation of eye color and its relationship to the sex of the fruit flies, Morgan assembled evidence that alleles are attached to specific chromosomes. Morgan found that his expected phenotypes in the F1 generation supported the hypothesis that genes for eye color were linked to the X chromosome. This finding had implications for how genes assort during meiosis. Several genes are linked to a single chromosome, and chromosomes are the particles separated during meiosis, not the individual alleles suggested by Mendel. Therefore, Hunt predicted not all alleles should independently assort, as predicted by Mendel. Different genes on the same chromosome should be linked. In other words, genes on the same chromosome should be dependently assorted, originally rejected by the 9:3 to 3:1 ratio of Corinne's and Mendel's dihybrid crosses. whereas genes on separate chromosomes should be independently assorted as predicted by Mendel. Linked genes violate independent assortment. In his experimentation, Morgan discovered another X-linked gene which affects fruit fly body color, with gray bodies as the dominant phenotype, expressed by X big B, and yellow bodies as the recessive phenotype, expressed by X little b. For females, phenotypic expression follows Mendel's principle of dominance, where homozygous dominant and heterozygous genotypes express the dominant phenotypes, gray bodies and red eyes. Recessive phenotypes, yellow body and white eyes, are expressed by two recessive alleles, X little e, and X little b, respectively. On the other hand, males only have one of these alleles, as they only have a single X chromosome. Whichever allele is present is expressed, where X big EY produces red eyes, X little EY produces white eyes, X big BY a gray body, and X little EY a yellow body. Since both alleles are on the same chromosome, X, we notate the genotypes accordingly. For example, a female with yellow body, a recessive phenotype, and white eyes, a recessive phenotype, would be expressed as X little b little e, X little b little e, while a male with red eyes, a dominant phenotype, and a gray body, a dominant phenotype, is expressed as X big B big E Y. Here are all the possible genotypic combinations of sex, eye color, and body color. Morgan expected the principle of independent assortment predicted by Mendel to be rejected based on his belief that alleles for both of these traits are attached to the same chromosome. He conducted a cross of dihybrid females, X big B big E, X little b little e, expressed as gray bodies and red eyes, with recessive males for both traits, X little b little e, Y, with yellow bodies and white eyes. If we assume gene linkage, the big B and big E alleles of X big B big E will stay connected during meiosis and fertilization. Likewise, the little b and little e alleles of X little b little e will remain linked. By completing a Punnett square, assuming gene linkage, or dependent assortment, Morgan predicted a phenotypic ratio of one gray body red-eyed female to one yellow body white-eyed female to one gray body red-eyed male to one yellow body white-eyed male. If you remove sex from the equation, he expected an even distribution 1-to-1 of gray-bodied red-eyed individuals to yellow-bodied white-eyed individuals. Gene linkage predicts no gray-bodied white-eyed individuals nor yellow-bodied red-eyed individuals would be present from this cross. Alternatively, if the alleles for body color and eye color were independently assorted as predicted by Mendel and Corin's, he wouldn't receive the 9-to-3 to 3-to-1 ratio since males only have one X chromosome. Rather, independent assortment predicts an even distribution 1-to-1 to 1-to-1 of the phenotype combinations. Results of his F2 generation did not support either of these alternative hypotheses. In his cross, he acquired 1,159 gray-bodied red-eyed flies, 1,017 yellow-body white-eyed flies, 17 gray-body white-eyed flies, and 12 yellow-body red-eyed flies. While the numbers are closer to the 1 gray-body red-eyed to 1 yellow-body white-eyed prediction of gene linkage and dependent assortment, the hypothesis predicted no gray-body white-eyed or yellow-body red-eyed combinations. While independent assortment did include these combinations, it predicted an even distribution of each combination. The truth was somewhere in between. But what was the mechanism? Again, the inner working of meiosis is responsible for Morgan's results. He suggested that alleles on the same chromosome remain linked during meiosis, but his results indicate that this is not always the case. Sometimes alleles on the same chromosome don't always stay together. Morgan proposed that during meiosis, paired homologous chromosomes can swap segments between them. He referred to these recombined chromosomes, part paternal, part maternal, as recombinant chromosomes. Further work by Morgan and colleagues supported this hypothesis, and they found that homologous chromosomes do in fact recombine during prophase 1 of meiosis. Morgan termed this process crossing over, and it involves the physical exchange of segments between homologous chromosomes. He concluded that genes connected to the same chromosome typically remain together, supporting dependent assortment. But occasionally there's an exchange of segments between maternal and paternal homologous chromosomes. This explains his result from the cross resulting in the F2 generation, which is predominantly a 1 dominant-dominant to 1 recessive-recessive ratio, with a rare emergence of dominant and recessive combinations in a single individual. As it turns out, Gregor Mendel was lucky. It turns out that the phenotypes he was observing just so happened to be on separate chromosomes. This allowed him to piece together a very simple model of inheritance, the particulate inheritance hypothesis, which includes the principle of independent assortment. Had some of his traits been on the same chromosome while others were on different chromosomes, he may have not been able to generate a working hypothesis to explain his results. Further research has revealed many other violations of Mendel's principles. In the early 1900s, Carl Corinz contributed to the reemergence of Mendelian principles of inheritance through extensive experimentation on a number of organisms. One plant he analyzed was the 4 o'clock, which have either red, pink, or white flowers. After he self-pollinated the red and white flowers, the resultant plants were pure lines represented by the P generation. According to the principle of dominance, he expected that the flowers of the F1 generation to either be all red or all white. To his surprise, all the hybrids of the F1 generation were pink. This result violated Mendel's principle of dominance and supported blending inheritance. Following the cross of the hybrids, the plants that emerged in the F2 generation did not express the 3-to-1 ratio that Mendelian genetics predicted. Rather, the phenotypic ratio was 1 red to 2 pink to 1 white flowers. He had discovered incomplete dominance. In this case, both alleles are expressed. Not one dominates over the other. In the case of a heterozygote, the flower color is a mixture of the phenotypic characteristics of the homozygotes. One of the underlying mechanisms Mendel used to explain his result was that each trait had one of 2 possibilities, such as white or purple flowers. In modern terminology, we would say that each genotype is composed of one of 2 alleles in homozygotes, or both alleles in heterozygotes. Investigation into human blood types illustrated some phenotypes that are expressed by more than 2 possibilities. In 1901, Austrian Karl Landsteiner discovered that humans have one of 3 blood types, A, B, or O. He discovered that blood transfusion by people with the same blood type did not lead to the destruction of blood cells of the recipient, which became an instrumental tool in the medical profession. Different alleles emerge via mutations during DNA replication, and they occur randomly. Therefore, it is possible for a gene to have more than 2 alleles, and some do. Human blood type is a well-known example. A gene known as I is responsible for the production of a specific polysaccharide attached to a glycoprotein found in the cellular membrane of red blood cells. The recessive allele i codes for the base polysaccharide. The alleles IA and IB code for the same base polysaccharide plus an additional sugar added to the end. IA codes for one sugar, while IB codes for a different sugar. 3 blood type genotypes are possible. IA, IB, IB, IA, IB, and I. Similar to Mendelian genetics, I is recessive for both IA and IB. Genotypes IA, I, and IB, I result in type A blood and type B blood respectively, where the recessive phenotype for type O blood is I. But what happens when both IA and IB alleles are present in the genotype? As it turns out, both alleles are activated, and blood cells with the genotype IA-IB produce both type A and type B glycoproteins. Neither dominates over the other. This double expression is known as codominance. When we think about inheritance in our own species, the principle of genetics discovered by Mendel are difficult to apply. Some of the most obvious phenotypes humans can detect seem to exist on a continuum, such as height, shoe size, and flexibility. Human skin color is a classic example. Ranging from dark brown to the lightest hues, skin color is determined by the presence of many substances, but most importantly, the pigment melanin. The ancestors of all humans originated near equatorial Africa about 1.2 million years ago. Being devoid of thick body hair, those people were assaulted by the sun's ultraviolet rays. Too much exposure to UV rays is a main determinant for skin cancer. Natural selection favored humans that had a built-in mechanism for reflecting UV rays, melanin. As humans migrated northward, they were dark-skinned, as revealed by recent DNA analyses of Cheddar Man in southern Britain. However, humans generate vitamin D from UV rays. The less melanin present in the skin, the more essential vitamin D could be generated. Hence, there was a selection pressure for lighter skin tone in more northern latitudes. As it turns out, human skin color is not discrete, either or, as predicted by Mendelian genetics. It is continuous. While the research is incomplete, we now know that skin color is determined by at least 7, likely more, different genes, or polygenic inheritance. Mendel and the rediscovery of his principles laid the foundation for modern genetic theory. But when we look closer at inheritance, every principle or law that was discovered in those early days has an exception. And those violations of genetic principles have paved the way for additional insight into the inner working of the cell.